Yes, hello everyone. Thank you very much for again for continuing this series and for including me in it for a second time. It's a great opportunity to speak to a wider audience without having to travel. Uh, so I have put together some slides, as you mentioned. I'm going to be speaking about our iSCAT work, but with a focus on one of the latest results, and that concerns iSCAT microscopy in live cells. So in a way, doing label-free 3D imaging in live cells. So let me then jump right in. I think the first reference to what we now call iSCAT was in this paper officially, where we showed that we could do detection and spectroscopy of gold nanoparticles. This was published in 2004. The context of this work was, in a way, should be understood as the tail of single molecule microscopy, where people were already quite aware that fluorescence has its limitations, and we're looking for alternatives. And about 2 years earlier, Michel had shown that you could do detection of very small gold nanoparticles using photothermal, and this was fluorescence-free. And we were excited about this work because it was also another version of fluorescence-free detection of very small particles. So it's really kind of in the context of trying to push the sensitivity limit without using fluorescence, but still using light. And gold nanoparticles have been used for a number of applications, especially in electron microscopy as label. So it was exciting to see how far you could go down to see gold nanoparticles using light. And we're also thinking of using gold nanoparticles as biological scattering label because they have plasma resonances. So that just kind of at the outset, a little bit of motivation or background of how we came up with developing this technique. But there was no biology. It was really just pure physics, trying to understand where the signal comes from. And in that paper, we understood that what is the underlying kind of quantity. The important quantity is the polarizability, and this is a textbook kind of result that you would have in your electromagnetic course. So the polarizability of a nano object is given by this expression, and polarizability itself is a prefactor that relates the incident field and the scattered field for a small object. So what we have here are the indices of refraction of the particle and the index of refraction of and of the medium. But what's in a way more important is this parameter volume in the polarizability. Let me get a laser pointer so that I can be more professional here. There we go. All right, and of course, polarizability is in a way the fundamental quantity that goes into the scattering cross-section and the absorption cross-section. So if you know polarizability of an object, you know almost everything about it in terms of its interaction with light. And of course, polarizability is a complex quantity because these dielectric functions or indices of refraction are complex, and you have to take that into account. And of course, these are wavelength dependent. So all of this is basic physics. Now, before I move on, I also want to emphasize that extinction is an interference phenomenon. So when you have a nanoparticle and you illuminate it, we just talked about the polarizability of this object and how much light is going to be scattered. But if you were to look right into the beam straight on and detect the power that you collect, then this power could be written or derived by considering the interference of the field that is scattered and the field that was incident. And the phases conspire in a way that you end up having less light going along the original K vector. And that's what is also known as the optical theorem. So what you really have is an interference phenomenon. The reason right, so this is kind of the equation that you would have for the intensity at the detector. Now, there are a number of techniques that are known in microscopy that use interference. Phase contrast, differential interference contrast microscopy, interference reflection microscopy, quantitative phase imaging, digital holography, and lots of other things that have their own names, but they all share the property that the signal that you get on your detector is the interference of two fields. One comes from the object, and the other one is, let's call that a reference. And it is important, if you want to realize, that also bright field microscopy is following the same equation as I showed you before, because what we measure in bright field microscopy is just extinction. Now, when you do dark field microscopy, you're detecting only the intensity that is scattered or emitted by the object without detecting the background. And when you're doing what we will call iSCAT is when you detect or analyze, treat the interference term. Now, what makes iSCAT a little different from these other methods, although the formula, the equation of motion is exactly the same thing, is that, and I'll be emphasizing this during the talk, is that we're talking about detecting scattering and not just any kind of a reflection or response of light, but scattering that is at the limit when particles are smaller than the wavelength of light. So scattering from nanostructures. That's kind of what we consider iSCAT. Otherwise, again, I emphasize that this formula is common to all of these different imaging methods. Now, in the way I usually define iSCAT, that just means that you can have any of these illumination and detection schemes, and it still would be iSCAT, because as long as you're looking at a nano object and detecting interference. So, for example, you could be focusing your light onto a nanoparticle and doing the standard extinction experiment. So that would be an iSCAT experiment. And you could be doing this in wide field mode, that is, you, for example, focus in the back focal plane of your objective so that you illuminate a large area, and then that beam interferes in transmission with the light that is scattered by the nano object, and then that way you can get an image instead of having a point detector. And you could also have your object sitting at an interface and therefore do this whole thing in a folded way in reflection instead of doing it in transmission. And then again, you can do this with a point detector, as I will be talking about in the latter part of the talk when we do confocal microscopy, and incidentally how we did it in the very first paper. Or you could be doing the same thing also using wide field, as has become very common recently. But you can also use a reference beam and interfere it with the scattered beam. And that's kind of what is done, for example, in holography or quantitative phase imaging. Again, all of these I consider iSCAT as long as you're looking at the scattered light and not other types of optical response. And of course, the terminology has evolved over almost 20 years. And so now I would like to clean up a little bit looking back. so that the community becomes a little more unified in understanding what really is happening and what's special about the imaging modality. Hello?
We can, Vahid, I can still hear you if you speak.
Yes, did you? I heard a very, very bad siren type of noise. Did you hear that too?
No, I didn't hear any siren, but you were cut for a while and then you are back. So I think everyone can hear you, but our chairman is not responding, so I'm not sure.
Yeah, everything is fine.
Okay, so you can continue.
I'm sorry about that. Yeah. Right. So where was I? Right. After having done, I gave you a little bit of theoretical background, and now I try to ramp up as quickly as possible to the actual topic of the talk today. On the way, I explained kind of how we got here. So after gold particles, what we realized was that okay, I think it's this video that doesn't want to speak. I heard that sound again. It's a very strange situation. I don't know what that comes from. So in this work, early on, we realized that we could actually apply iSCAT to viruses and looked at diffusion of viruses. That would be in this movie, but it looks like I can't play that. Then we went to get better sensitivity and detection sensitivity, and we moved towards detecting individual proteins. And in this work that was published in 2014, we showed that we could apply iSCAT and detect very small proteins down to 65 kilodalton. And we also showed that the signal that we get, if you remember, because the signal is proportional to the volume, this signal actually scales with the mass of the protein, because it turns out that the density of protein matter is pretty much the same regardless of what kind of protein you have. So the polarizability is not only proportional to the volume, but also proportional to the mass. So this is what we observed in that work within the accuracy that we had. More recently, we've improved the sensitivity from 65 kilodalton down to just below 10 kilodalton by learning how to improve our signal to noise ratio. And there we've exploited some of the new machine learning techniques. This is not something that I'm going to elaborate on today, but you can read this paper that came out earlier this year in Nature Methods. And now detection therefore goes now to about 10 kilodalton, and it's been pointed out, and after we've shown that the protein mass is something that you can have access to via iSCAT, the group of Philip Kukura very nicely showed that you can really build a ladder and calibrate your setup and use this as a quantitative way of determining masses. And in particular, they showed, for example, that you could distinguish between dimers and monomers and dimers and trimers, for example, in a sample. And biochemists seem to be very interested in this, and they've moved on to establish a company. So this has become now commercialized. This was in 2018. And in the same year, we tried to apply the same idea to studying secretion from a live cell. So cells usually secrete proteins and nanoparticles and such. And in this particular case, we knew that this cell was supposed to secrete IgG. And what you have here is a video of proteins that are being secreted. When we put the field of view a few microns away from the surface of the cell or from the cell, and you just monitor in real time how proteins land. What you see here at the very end, this is after more than half an hour, this cell was lysed because the pH had gone out of control. But along the way, we could establish histograms where you can show, for example, the contrast of the proteins that you've detected, the iSCAT contrast, and you see how frequent which contrast is. And again, because the contrast is the same thing as mass, you can figure out how much of what kind of protein was secreted. And in that particular case, we could see that our IgG is coming out, but there's also other things that are being secreted. Another thing that is fairly new, but it goes along the same direction, is the interest in sizing nanoparticles and also establishing their refractive index. So this is something that has been available commercially using so-called NTA, nanoparticle tracking analysis. And what these commercial devices do is they have a suspension and they illuminate that in a dark field mode and basically track individual nanoparticles in dark field microscopy. And by looking at the trajectories, they can extract the diffusion coefficient. And from the diffusion coefficient, you can extract the size of the particle. But if you remember, dark field microscopy is proportional to the volume of the particle, and the signal becomes very small very quickly when the particle becomes small. What we realized is that iSCAT has this advantage that we can detect smaller particles. So we showed that if you do the same type of NTA but in interferometric mode, and therefore the name INTA, we can basically go much further in terms of sensitivity. So here is a quick overview of the performance. Another method that is commercially available and very old is dynamic light scattering. So here is benchmarking. You have some particles that are mixtures of 30 nanometer gold, 20 nanometer gold, and 15 nanometer gold. And in DLS, you would get a distribution that looks like that. In NTA, you would also get a distribution that is not separating these. But in INTA, we could separate these different clouds very clearly. And here are a few other examples of applications where we've mixed different types of very small nanoparticles and could show that nevertheless you can decipher them. I'm not going to dwell on this, but you can read the paper if you're interested in this. This is right now very exciting because there is a lot of interest also in biology of detecting small nanoparticles. By now, everyone knows that it's important to detect viruses, but a field that is particularly exciting right now and active is detecting extracellular vesicles, the so-called EVs, because these are supposed to be, they're hypothesized to be good disease markers. These are basically things like liposomes or small SUVs that have proteins in their shell or inside. They could have RNA, they could have proteins, and so analyzing this is very important. So now I come to the main part of the talk, or the second part of the talk, which carries the title. In this recent work, we showed that if we do confocal iSCAT, we can go inside the cells and get an extremely good signal. It's to be remembered that because iSCAT is a coherent imaging method, prior to this work, we and everyone else was bothered by the fact that you have a lot of speckle when you look at something as busy as a cell. And what we showed in this work is that by doing this in confocal mode, you can get around a lot of the problems. So in wide field iSCAT, you'd be illuminating a large area and then interfering that, for example, in reflection with the light that is scattered by this particle. Let me get my laser pointer again. Right, and what happens there is that you're basically interfering something like a spherical wave with a plane wave, and that leads to very strong side rings. When you do this confocally, your back reflected light and your scattered light are much better phase matched or matched in wavefront, so that the point spread function is much more confined. It doesn't have these rings. It doesn't extend as much. And that is one of the reasons why confocal iSCAT doesn't give you nearly as much speckle. But of course, the other reason is that you have a pinhole, and the same way that the pinhole selects only a small volume from your sample in, for example, fluorescence confocal microscopy, the same thing happens in iSCAT microscopy, so that you don't have as much background that would interfere with each other and with your field to give you this speckle. So here are examples of two-dimensional PSF and the axial PSF of these different modalities. This is a standard confocal microscope that you're used to, and this is the confocal iSCAT version. You see here that it's richer than in the normal intensity measurement, because there's a phase change in the focus going from bright to dark, and I'll come to that in a few slides. All right, so I'm now going to show you a whole bunch of images and videos that give you a flavor of how well this works. And if you're interested in this, then I refer you to the paper, because there's a lot more detailed discussion in the paper and in supplementary materials. So one of the things that I would like to discuss is detecting the endoplasmic reticulum and microtubule in a label-free fashion. So in this picture, before I run the movie, I'll just tell you already that the darker tubules, or the darker kind of lines, are the ER, but you also recognize weaker ones, the fainter ones, and those happen to be microtubules, and I will show you why we're sure of that. So let's run the, that's what I was worried about. I think I just have a defected file. I'm sorry about that. All right, this is a zoom into a similar type of image. Let me just go back here. You appreciate here the size of the scan that we have. This is the periphery of the cell. So you clearly see that over a large region, we're seeing all of these things without having to label them. Here is the a zoom into that region, and again, the dark regions are the ER, and then once in a while there is a, if I run the movie long enough, you will see faint ones that are the there it was, that are the microtubules. Now here is, for example, a microtubule, and the ER runs on the microtubule. This is a phenomenon that is known, so we can watch all of these things without having to label this with fluorescence. And of course, once you do that, you can also analyze the dynamics of all of these things. This is a map here. I seem to be stuck. Do you hear me?
Yeah, now it's fine. You were out for 10 seconds.
Yeah, now it's fine.
Okay, but my computer is stuck. Listen, I apologize for this, but I'll go back to my laptop because I tried to do a better.
Yeah, it's fine.
Everything, but I'm not able to change change slides anymore. My computer is frozen. Let me come in with the other one.
Recording in progress.
Do you hear me now?
Yes, we hear you.
Okay. You probably have a different sound quality because I'm on a different device. But now I have to get back on. Okay, so now I can watch, I can run this video. And so
Sorry, I think you didn't share the presentation.
sorry, what happened?
We do not see your
Oh.
slides. Yeah.
I thought I did.
Yeah, now it works.
Okay, sorry about that. Right, all right. Take number 5. So here's the video that I wanted to show you. And this makes this kind of imaging very exciting because you can see lots of things that are happening without having to label them. And these include lots of small vesicles and things that we have then tried to identify. And I will show you a little bit of that in the next slides. So I showed you this video already, and there was the running of an ER on microtubule and the fact that we could analyze the motion of the ER I talked about. And now one thing that happens when you do this label-free imaging is that, of course, you don't have the great advantage of chemical specificity that you have in fluorescence. But what you can do nowadays is you can train your computer via a neural network to learn the general features of a certain type of structure that you have, and then try to identify that in your videos. So we call that, now you could call that computational specificity or in silico specificity. So here is an example of a fluorescence image of the ER. And here's an example of the iSCAT image of the same ER. The iSCAT data that I'm showing you were all recorded on a confocal microscope that is a standard fluorescence confocal microscope. And because the two channels are two different wavelengths, we can have access to both of these signals at any given time. So now a network was trained on the fluorescence data, and when it was then applied to the confocal image, it could recognize to better than 92%, if I remember correctly, the ER tubules. Now, I showed you before that I claimed that one thing was ER and the other one is microtubule. And we know this because we can, of course, take advantage of fluorescence. And here is an example where you see that, again, there's a faint line that is the microtubule because we know how we've labeled the microtubule. And there's a darker one that is part of the ER because we've labeled that with a different dye. Another exciting thing here is that because iSCAT is an interferometric method, we have very good information about the third dimension, much better than the usual confocal axial resolution. And that has to do with the fact that when a structure or nanoparticle, nanostructure changes its height, you end up having a phase change, and that phase change is encoded in the contrast, and therefore you can extract your three-dimensional structure. So here is just an example of a part of this ER that you can see here in 2D. We can also visualize that in 3D. Another interesting thing when you apply this was that this method to different cellular type of events. There are also ER sheets. So there's the endoplasmic reticulum tubules that we've seen, but there are also parts that form something like a carpet. And it had been hypothesized that this sheet, kind of like a drum piece, might vibrate in different ways. And we think that we actually have seen this now using the great axial sensitivity of iSCAT. So here is a part of a cell that we've imaged, and what you see here is a section of the ER sheet. You see that the contrast has bright and dark parts, and again, that's because these are slightly at different heights, and you can analyze that to learn about the three-dimensional topography of this sheet. And by looking at over time, then you can figure out how often they move up and down. And this picture here is a map of how often these things, different parts, have been going up and down. Again, a lot more details are in the paper. But if you were to look at the fluorescence image of the same region, you wouldn't be able to say anything about it because, of course, the fluorescence appears to be fairly homogeneous. Another application that I'd like to show is clathrin endocytosis. So here's an iSCAT image. Again, you see that there are parts that are dark or parts that are bright. That has to do with this phase difference. But then you also have a whole bunch of small dots or black spots. And if you just see a black spot, you know that it's probably a nanoparticle, but you don't know exactly what kind. But knowing your application and a little bit of cell biology, you can guess. And in this case, we ruled out a few things and we realized that these must be clathrin pits. So then they were labeled, and that confirmed that these were all clathrin. And so here's now the video of it. These are these dark spots that you see. If you look at one of them, then you see that that one and this one, the contrast changes. And the fact that the contrast changes shows that these are now not just moving, jittering in 2D, which is shown, for example, here. This is now a slight trajectory of each one of these clathrin pits in 2D, but we can also extract a 3D trajectory of it because by analyzing the contrast, you know how far it went up or down. You can do the same thing to a virus. So in this example, we've put down a couple of virus particles. These were actually COVID-like COVID virus-like particles. And so again, they're fluorescently labeled just for us to do our benchmarking. But then afterwards, we can chase them and track them in iSCAT and figure out their 3D trajectory that teaches us something about the way the virus is interacting with the cell membrane. Now, to close, I want to emphasize that what we now again call iSCAT is something that in principle has been around for a long time. already in the early days of confocal imaging, people realized that they would get very interesting contrast by looking at the reflected signal. For example, in the group of Amos, they had this paper already in 1987. They call that reflection interference contrast. But the way the arrangement of the microscope is put together is almost identical to what we do today in iSCAT. Okay, so they're looking at things, this scale bar is 100 microns, so they're first looking at very, very large objects. So in a way, reflection is justified because they're looking at light that is reflected from objects that are much larger than the wavelength of light. A couple of years later, they reported that they could also see microtubules. These are, of course, not inside the cell, but on a cover glass. And again, they call that confocal reflection interference microscopy. But today we know that this is not the right way of thinking about this, because a microtubule doesn't really reflect light, but it scatters light, because the cross-section of a microtubule is in diameter only something of the order of 20-25 nanometers. So this is a nanoscopic, if you want, a nanowire or a nanorod. And you have to understand that signal, again, looking at polarizability. Of course, this is a polarizability of a sphere, but you can derive the polarizability also for an elongated object, and it would just be a form factor that goes in here. But what remains is that polarizability has a unit of volume, and so the volume is an important factor. Now, if I were to estimate the volume of something like a microtubule, if I take the basically a cross section that is 20 by 20 nanometer, and take the length of it to be about 200 nanometer. That's the length that matters to me, because that's the part that is in a confocal, that is in a diffraction limited spot. So I'm getting light signal from about 200 nanometers of it in one diffraction limited spot. Then I would have a volume, this is an estimate, of about 80,000 nanometer.
Sorry, 2 more minutes.
I should be done earlier. And if I were to look at the volume of a small protein, this would be only about 64 cubic nanometer. So we're talking about 1,000 times smaller object, order of magnitude. So this is really the contribution of what we can do today compared to what was done 20, 30 years ago. And it's really been important to understand that this is a scattering signal and not reflection, to be able to push the limits. And of course, there is now potential for not just looking at the polarizability of a spherical particle, but bringing in shape into this. All right, I'd like to thank all the people who've contributed to the development of iSCAT from the very early days. Thomas Kalkbrenner was the very first person who ever saw an iSCAT signal in the lab. We were still at the University of Constance. Again, over the years in Zurich and now in Erlangen, a number of bright people have contributed to our work in different ways, and I'd like to thank them all for that. We do have openings for new group members at the postdoctoral level, so if you're interested, please let me know, in particular in the context of the research at the interface between fundamental physics and fundamental medical research. And with that, I'd like to close, and I apologize for some technical difficulties.
Yeah, thank you very much. It was wonderful. I already see a lot of questions, so I will start asking, but just want to let everyone know at the end of our webinar, we will have a, let's say, 20 or 30 minutes open discussion. So we can also still discuss at that point. If I hope the Professor Sandoghdar will be there. I will start, there is already a question in chat. So let's start with Guillaume question. So what is asked, what do you call the ER? Aren't they also chains of mitochondria?
No, the ER is a different organelle than mitochondria. The ER is much weaker in terms of signal. These are tubules that are about 100 nanometers wide, but they're not as dense as they're not as densely built as microtubules. They're wider in terms of diameter, and they're fluffier, if you want.
So maybe to clarify,
yet...
I'm sorry. So maybe to clarify my question, I was wondering if what you see, so you say it is the ER, but can be also mitochondria, because the image you are showing, it is exactly what we see with wavefront microscopy. So the mitochondria are very clearly visible on our images, and they look exactly what you see. So I was wondering if there is also a contribution of the mitochondria on the images you see.
Right. So, I mean, we clearly also see mitochondria. Again, you get a sense of the strength of the signal. The mitochondria give much larger signal than the ER. So by knowing your microscope, then you can kind of judge. But we have labeled all of these as well in fluorescence. So we benchmark. So what I call ER is really ER. And but we can also watch the mitochondria. Now, if you have mitochondria and ER in one image, again, you can identify both of them, and it's true that their shapes might look very similar, but their signal strength would be different.
Okay, thank you.
Okay, we go to second question from Godai. Comparing different methods for 3D imaging, does that mean that for the slow objects like cells, the confocal method gives better quality? However, for fast object as gold nanoparticle, one should still operate in wide field. So his question is, if it's for fast object and gold nanoparticle, one use wide field?
Right. So, I mean, it's true that confocal microscopy is always limited. Well, it's not always, but it's quite often limited by the speed of the scanners, because it's a mechanical scanning that is done. At the end of the day, you have to scan your beam, and that it cannot be done as fast as we can run a camera. So it's true that also in our paper, we show that when we do the virus tracking, when we want to really be very fast, then we switch between confocal and wide field. But again, you have to know your application. The wide field, we have demonstrated as well that you can track nanoparticles and proteins and all of that tagged with nanoparticles, but you have to work a lot harder to get rid of the background speckle than in confocal.
Okay, question from Ming Seng. Very nice talk. Compared with wide field iSCAT, in addition to get rid of the dinging artifacts, does confocal iSCAT improve the sensitivity as well?
So the sensitivity depends on what we mean. I mean, the sensitivity in iSCAT is quite often limited by background fluctuations. And in that way, yes, confocal iSCAT could improve the signal-to-noise ratio and therefore sensitivity if you can get rid of your background and therefore its background fluctuation. But again, the applications are quite different. I think there are some cases where you can't, for example, for 3D sectioning, you just can't use wide field, although it's much faster. And for fast things, you can't use confocal because it's just not fast enough.
Okay, question from Paulo. Nice talk. Can you comment on the requirement of the distance in G between the object of interest and the cover slip, providing the reflected reference in confocal iSCAT? In other words, how deep can you image?
Yeah, that's also a good question that we discussed in the paper, and it's supplementary. Without doing anything, you can easily go as deep as 3-4 microns. And if you want to go deeper, then you have to replace the reflected light from the interface by an external beam, because your confocal pinhole ends up discriminating against the light that is reflected from the interface, because it's no longer parallel, and therefore it gets cut off at some point. So that's something that we have done, and but it makes the setup a little more complicated.
I think Michelle, you had a questions or similar question? Okay, I think you are mute, Michelle.
I just removed my mute. That was my question. It's answered. Thank you.
Okay, I think the last question from Professor Kobayashi: how is the fastest frame time?
So in the images that we've shown you, we're imaging at a few hertz to a few 10 hertz in a typical kind of confocal image. But again, that's something that you can choose. If you choose to scan only a very small region, you can go faster effectively. And we're limited by the scanner of our confocal microscope. So one thing to also remember, compared to fluorescence, iSCAT gives you a lot more signal. So you're really not limited. You could integrate very, very short time if your scanner allowed you. And you can imagine that, if you were to build your own setup and had an acousto-optic kind of scanner, then you could be a lot faster.